So I would like to thank the organizer for the opportunity to speak at this fantastic meeting. So really it's been an inspiring day, a series of talks with filled with discoveries, optimism, and somehow I'm entrusted with a task, right? So the formidable task to basically bring to a full completion, right? So no pressure. Great. So this, So really today, and also I was told that this, for average audience, they have the attention span of 2 slides. Right, so you're gonna witness my best effort to make my first 2 slides count on this topic of this SYNGAP gene modulation therapy and biology. So first, I would really want to drive home this idea of gene modulation therapy. Right, so really think of it as a dimmer switch you may find in your hotel room. Once you find it, you can turn it up or down, right? So it's so you can change output of its targets. Say it's a light bulb or it's like AC unit. So, and every single gene, including Syngap, has built-in dimmer switch. So to make sure it's expressed in the right cell, right, at the right time and in the right amount. Right. So this, so idea really is similar to in a normal cell. So there's 2 alleles. Think of it as 2 light bulbs lighting. Right, so in the Syngap deficiency syndrome case, so one allele gets decommissioned, so the 1 light bulb went out, so not enough light, therefore haploid insufficiency. So really now keep in mind that there's still a light bulb that's intact, right? So it's a wild type allele. So can we find that dimmer switch and use that therapy to engage it, right? So turn on this remaining light bulb, make it brighten up. So this type of gene modulation therapy is fully titratable and is reversible. So think of it as you can always take more drug or less drug, or if adverse event happens, you can stop taking drug. So it's have the precision at the same time have adaptability in a clinical setting. So this, and also use slides to basically summarize, like give an outline of this talk. So this, so it starts off as in our effort to find really the next generation of anti-seizure medication that engages the brain's endogenous brake system, that's a garbage inhibition, without causing the sedative effects of this benzodiazepine. So we, through unbiased drug screening, we found a small molecule drug inhibits the FLT3 kinase. And this, some of these drugs can enter the brain, right? So it can cross blood-brain barrier through either oral route or through IP route, and can engage the target, change the gene expression in the brain, and reduce seizure susceptibility without causing sedative side effects. And with the support from the Syngap Research Fund, we found this occur SYNGAP1. So we found this increases Syngap expression in the brain, and it has a quite remarkable long window of efficacy. Then we drill down to understand how this drug works. So for that, we really use a functional genomic approach to really understand the transcription programs, that's the transcription factors involved, engaged by this drug. And that works on Syngap. So from this, we found actually, to our surprise, quite a few of this inflammation and immune-related pathway, stress response pathway. So those transcription factors, they are normally in the brain, they suppress the expression of Syngap, and our drug will relieve that suppression. Right. So this also will talk a bit about, we follow the whole biogenesis, basically the whole life cycle of Syngap, from DNA to RNA and RNA to protein. So we share some of that results as well. And then with these insights, we went into drug design, where some bespoke therapies. So in this case, we use antisense oligo, this modality, but instead of targeting Syngap, we target the FLT3, this kinase. So we optimize a couple of candidates, and we optimize the dosing, how much we give to mice, and map the time course efficacy. And happy to share with you, we found some candidates able to increase the Syngap expression in vivo for a very prolonged period of time, and also can reduce the seizure susceptibility using the PTZ challenge. Right. So I do have more slides after this. All right, so start with this FLT3. So we found this out of unbiased drug screening. So basically, what it does is a receptor tyrosine kinase on the membrane. So it transduces signal, and it studied a lot in the leukemia, basically in the blood cell production and the blood cell overproduction scenario. So we're the first one to look at this in the brain. So what we found in the brain, on the left side, you can see the panel. So it's those donuts, those red donuts, those are FLT3 staining. So this co-locus is pretty much perfect with the neuron. Basically, it's expressed very enriched in neurons in the brain. Right? So this means once a drug enters the brain, it's going to engage the neurons, but not other cells which don't have a receptor. And what we found is overall, if you look at all the genes response to a drug, it increased the expression of synaptic ion channel genes, at the same time reduce the neuroinflammation type of genes. So this gene set enrichment analysis plots on the right indicates. And we went further to look at the autism risk genes. Right. So in this case, we subgroup alternate risk genes into different categories, functional categories, and we found similar to the full gene analysis, the alternate risk gene has to do with synapses, SYNGAP included, and also with ion channel. Those are ones stimulated by the FLT3 inhibition. And importantly, SYNGAP is one of those hits. Right, so it's really across different human and mouse cells and also across different treatment conditions. It's quite consistent. Right. So this. So I mentioned this drug is this our top hit drug, KW249, really is orally bioavailable. So this was actually used in leukemia clinical trials through the oral administration. So in mice, we were able to show that the drug really either you give by lavage or give by injection, it can enter the bloodstream, can enter the brain, right, engage the target, and with a half-life in the blood about 3 hours. And then it's really, in collaboration with Jay McGuire's group at Tufts, we did the EEG recording, look at this, their response to kinetic acid-induced seizure. We can see pre-treatment mouse with the drug a day later, right? We can see the drug disappeared already, but this gene expression stimulation was still persists. So it really confers quite substantial protection against Ka-induced seizure. Right. So this, and we'll also look at this side effect because we really think this pathway engages the basic chloride homeostasis in the neuron. So that's reinforces garbage inhibition. So this, and we look at this, we use two tests. One is called open field test, look at how mouse behavior, movements, and a rotarod, look at the motor capability. So overall the message is, so different from benzodiazepine, which have very strong sedative effects. The mouse basically sits around, don't move much. So our drug, either 30 minutes acutely or 24 hours after the gene induction, so really don't have any obvious side effects, right, comparing to the control. So I mentioned SYNGAP, right? It changes SYNGAP. What about this? What about SYNGAP in this mice receiving the drug? So what we found, so we basically have a cohort of mice. We inject them with the drug, just one dose, and we collect them at different time points, right? Either DMSO injected or KW249 injected, different time points, each names of mice. So what you can see is overall, so it's actually, it takes a couple of days for the SYNGAP expression to go up in the brain, but it persists all the way until 2 weeks. So it's quite like, we find it quite surprising, right? So we did multiple repeats to really consolidate this finding. And putting this in context, our NAPS another favorite gene called KCC2. So this, so what we found is KCC2 follows what we would predict at the beginning. So the drug, the red line indicates the drug was out half-life about 3 hours. The KCC2 goes up about 24 hours and stays about a day, right? It goes down. That's like a normal, right? So the protein turning over. In contrast, SYNGAP really in the same brain, in the same group of mice. So it's really take a couple of days to go up, but then it persists for quite a long period of time. So really this suggests that this the same drug, right? So in treating different, potentially treating different genetic condition, we can use different dosing, right? So to use that to achieve more precision and also to even reduce the potential side effects and any potential side effects. So then we went down even further. We tried to understand how this drug works. Right? So it's a FLT3 kinase. It's on the membrane. So this, so the gene transcription is supposedly in the nucleus. What's the connection there? So this, the SYNGAP project really benefited from our KCC2 project, which we conducted a large-scale genetic screen. The idea really is from our sequencing results, we found the transcription factor that's regulated by this drug treatment, and then we use CRISPR to knock them down one by one in the cultured neuron, right? So then you use either KCC2 or SYNGAP expression as a readout. So for KCC2, we screen a whole bunch of transcription factor. For SYNGAP, we use the winners from the first screen, right? So we only basically see whether those TFs would work on the SYNGAP. So here, so for this SYNGAP study, we particularly included the SYNGAP targeting, guide gate. So you can see it on the second bar. So the CRISPR works, the antibody works. So we found among the transcription factor that work on the KCA2, a fraction of them works on SYNGAP. And it's very interesting because these genes actually, if you look developmentally, they turn on more or less at the same time. Actually, it's kind of like they have a, I would say maybe SYNGAP is kind of like KCA2 more kind of sharper increase, but overall by like when it's not maturation, they're very high level. But they both respond to the same drug, but it's actually through not complete overlapping pathways. So we find it quite interesting. And what are those hits? a couple of this heat transcription factor in this pathway of this cytokine signaling and immune response, right? So like JAK STAT pathway and ETS, right? So this really, I would say, last year I was at this reception talking with some parents, and they told me that this some patients gets worse when they're in fever. I find it very surprising because in autism in general, people think fever has a sort of protective effect, right? So, but this might be part of like help us to understand this, like immune response, right? So, or to like a disease-specific immune response. And some other hits in this fourth stream pathway. So if you're neuroscience aficionado, you might recognize those as driver activity-dependent gene expression program. But fourth stream is actually widely expressed in the body, and in a non-neuronal cell, they are function as a stress response pathway. Right, and the RUNX1 is the same thing. Basically, similarly, they converge with the fourth string on some of the pathways. Right, so this really, I think this by understanding, this signaling cascades really give us a lot of biology about how the Syngap is, what the dimmer switch is, right? So in the Syngap gene, endogenous in the neurons. Right, so when I step further to look at the biogenesis of this of Syngap protein, because our hypothesis like this. So this you have this drug induction very short period of time, and then you have very long period of protein. Either protein is very stable, or somehow the RNA is very stable, right? So in this case, so we want to look at where this protein is produced. So I was very happy to see this talk in the morning. So I can show some like similar results, but we came up with a different approach. So we use this called pure myosin proximity labeling. So the idea is you add the pure myosin to your culture for 10 minutes, for a very short period of time. You label all the protein being actively produced in that period of time, and then use one antibody against pure myosin, another antibody against Syngap, and you can visualize where in the neuron are the Syngap produced in the last 10 minutes. So what we found is like, so if you stay for total Syngap and use this proximity labeling, look at Syngap produced in the last 10 minutes, right? So before the cell gets fixed, there's essentially 100% overlap, right? So this in this putative synaptic sites, and this showing this Pearson correlation. So it's really it's very actively produced. Pretty much at all the synaptic sites. This is a DFA21. When your spine is established, the cells are very synaptic connected. And we found a very similar thing even in the 7 days, even at the very early stage. So at the 7 days, there's no dendritic spine, right? So the synapse like barely forming, but yet those Syngap already clustered and is actively transcribed in those, translated rather, in those synaptic sites. So I think this really, so I would like to think, and our KCA2 is opposite. Our KCA2, basically we do this, our lab have two favorite genes, KCA2 and Syngap, we contrast this. KCA2 is all pretty much like the iPhone in your pocket, it's produced in central factory, shipped around. Syngap, on the other hand, we would like to think as a kind of like a farmer's market, right? So it's produced locally and consumed locally at the synaptic site. So what does this, so I would feel a few of the thoughts on this, what this means for therapy development to understand biology. So first, really our model really is that the protein production is at synapses, so the RNA must be trafficked. Right? Because RNA is produced at nucleus and it must be trafficked at the synaptic site. So maybe there's some isoform specific, like this affinity, right? So go to this RNA, maybe the different stability at the synapse. So I think that is a rich field to explore. And also, because there's continuous synthesis, that also means there must be an active degradation at synaptic site, right? So in that case, maybe we can shift the degradation pathway, right? So proteasome pathway, use that to change the inventory, if you may, like of a Syngap synapse. And also, I was quite fascinated by this early, very early clustering of Syngap at synapses. I wonder whether it's play a role in the synaptogenesis in addition to its role in the plasticity. Just a couple of thoughts in this area. All right, great. So last but not least, we're going to talk about ASO, right? So about the designer drug using what we learned. So this. So can summarize a lot of our optimization effort. So this we design multiple ASO candidates because there's many ways to skin a cat. Right? So Syngap is a very long transcripts and many ways you can use to knock it down. Some work better than others. So we tried multiple candidates and we optimize this intraventricular dosing. So it's just one injection, right? So into one side of the ventricle, and they also really permeates the brain and we collect sample from the other side to do the analysis, right? And then we assess efficacy over time, right? So use brain tissue extraction and also look at this anti-seizure efficacy. Right, so I'll quickly show you some data. One is like, with our top candidates, so in a dose-dependent manner, we can find like a substantial increase in the Syngap protein level, right, at 2 months after a single injection. Right? So we're quite happy to see this. And and we followed it up by this PTZ challenge. So this using so idea is you start with ASO injection, and then you then like a month later you challenge this mice with a high dose of PTZ. In this case, it's kind of similar to what Rick mentioned earlier, but we use a higher dose to induce. So this. Excuse me, and then use a video recording and a Rasen scale, like just basically. So what you're looking at 2 hours of this of the recording, and this every 10 seconds there's a beam, and so we visualize, and then like look at seizure behavior. The red means severe seizure, white means no seizure. So you can see overall, one of our candidates, right? So this really, you see a lot of blue and white, not much red. Whereas like another candidates, which are the protein levels look somewhat similar, but somehow this don't have as much of the protective effect in terms of function. So we're still sorting out what's like this. We're still sorting out the details of this. But this I'm happy to report we have some ASOs that's able to confer some anti-seizure efficacy. We're happy to we're actually actively working more in this space. All right, so good. Is a short time I have, and I will have a shout out to my students' poster. So now we understand. So this really is about this understand non-coding variants. So what regulates this expression of the Syngap, right? So in addition to this protein coding variants, there are many more non-coding variants in the intron region. So so now we understand the so-called trans elements, whether the transcription factor that regulates Syngap. So we try to find the cis elements, right? So what are the DNA binding sites? What are the important sites that regulate expression? So Lanxin, a student in my lab briefly take on the project. So here just quick summary. So she looked into neuronal epigenome data, transcription factor binding, put in evolution perspective, like genomic data from patients and normal people, and add some AI spice. So end up with this pipeline that's able to prioritize the variants. Boil it down from 22,000, now prioritize to 20. So which we can use that to do DNA editing in the human neurons. Use that to validate those predictions. So check out the poster if you haven't done so. Great. So this so check out her poster, right? Of course. Yeah. Great. So so we're taking this. So most of the study we show this in the wild type mice, right? Wild type mice, wild type cultured neuron. So this we really actively using this to rescue Syngap haploinsufficiency insufficiency in the cultured neuron, human neuron, and also in the mouse model. So for that, so this you're looking at my R1 Ames page, right? So look at this. So that's really we're actively pursuing this. Right, great. So this are my labs folks. So they did the work. I got to talk about it. Yeah, so really thank you for the for the Cure Syngap for this platform and for the support. Thank you. I'm ready for questions.